Hello, and welcome to A Scientist Walks Into a Bar. This podcast presents recordings from a series of events called Science on Tap that are held in Portland, Oregon and Vancouver, Washington. In this episode, we'll hear Dr. Thomas Seeley talk about his book, The Lives of Bees, the Untold Story of the Honeybee in the Wild. Tom is a professor of biology at Cornell University, and he's been studying bees for decades. He's also written several books about bees, and we'll include links to all of those in the episode notes. In his talk, Tom discusses the idea that looking at the behaviors and adaptations of wild honeybees could give beekeepers some ideas for how to fight back against colony collapse. He spoke in Portland at the Alberta Rose Theatre while on his book tour in May 2019. Quick note on the talk. We do trivia at all of our events, but it doesn't make for great audio. Tom mentions the trivia a couple times during the talk, so we wanted you to know what he was talking about. With that, here we go.
Let's give it up to Tom Seeley.
Thank you for coming out. Thanks to you folks. What I'm going to share with you tonight is this story. It's really an untold story, or at least very little told story, about how the familiar honeybee, Apis mellifera, lives in the wild, and why that's important and useful to us today as we deal with the issues of high levels of colony mortality because of the introduced parasite, which is so aptly named Varroa destructor. So that's what we're going to be looking at tonight. But just to remind us to put it all in context, let's go back and look at why E.O. Wilson calls the honeybee humanity's greatest friend among the insects. There are really two reasons. One is they are the makers of honey, and we really like honey. And not just the Hadza, and by the way, those Hadza have very huge dental problems because of the honey they eat, but that's not so good. But it's a time of year when it's hard to get game. There's rain, the grass is tall, it's hard to be a hunter, except to be a honey hunter. So, and here is that drawing that we saw in the quiz from the Cave of the Spiders. And the thing I want to highlight about these images is that these images they emphasize just how important honey has been to humans for thousands of years. Because over on here's another drawing. Here there are 5 individuals climbing up this ladder to get to a nest up here, and 2 of them are falling off. And that's a big distance down. So it shows you the dangers to which people would go to get this wonderful food. So, and then here, of course, is a more modern image of what honey looks like. How many of you are beekeepers and have had this experience of seeing honey flowing from the extractor? Oh, good, great, yeah. So you know. How many of you have gotten sick after extracting honey because you're always taking some? So yeah, that's one of the reasons they're our greatest friend among the insects, honeybees are. The second is that they are our single most important pollinator species. There was a recent study done where 49 scientists worldwide got together. These are all experts on different bees. They figured out for each of our human crops, how much each bee species that is known to pollinate the various crops that we have, how much of that pollination for each crop was done by which and which species. And when you add up all the numbers, the honeybee, Apis mellifera, does 47% of all crop pollination services worldwide. So that's a lot for just one species. It means it's as much as all of the other hundreds of species combined. That's not to put down the other species, because the honeybee is not always a good pollinator for every plant, but it is a very important pollinator. And I want to share with you now probably my favorite picture of a honeybee. The bee is carrying the load of nectar in its abdomen, where it's translucent, and it's got the loads of pollen on her hind legs. And what I really like is the way she's got her legs tucked underneath her. It's very cute. It's for her streamlining, of course. So that's our hero for tonight, doing both the honey making and the pollinating. And this reflects back or links back to one of the slides or the questions in the talk. Are the movable frame hive. And because this bee is so important to us, we have developed over thousands of years more and more sophisticated hives. And this is the latest, well, maybe not the latest, but it's certainly the greatest hive that's been invented. And this was by this gentleman, Lorenzo Lorraine Langstroth. And those of you who are beekeepers know that Yeah, these boxes, they don't look like much from the outside, but if you look at them from the inside, you'll realize that there's these wooden frames in which the bees build their combs, and they're very carefully designed, so there's a thing called a bee space on all the sides, so the bees don't attach the wooden frames to the sides of the boxes. That bee space was, and this invention of this movable frame hive, that was Langstraw's big invention. And it's really nice. You can just pull these combs, these frames of comb out of the hive like cleanly and without just damaging the nest or the bees, much like taking the folders out of a filing cabinet. So that's one of the things that we do. There is another reflection of how important these bees are to us. We've made these special hives. And so we've got a lot of the bees living in our hives. As we'll see, it's not the majority of bees, but colonies, but it's a large number. And by doing so, we gain a lot of control over their lives. We encourage them to produce more honey than they would in nature because we give them a lot of space in which to store honey. We can do things like inhibit the swarming or colony fissioning. And we can just generally make it possible to go into their hives and do things that we want to do, such as take out the honey, divide colonies, and the like. But even though we've got all of those means to manipulate the colonies, we really haven't changed at all the behavior of the bees. They still do what they want. They go out. We don't tell them where to go. We don't tell them what to do when they get outside the hive. We don't tell them what to do inside the hive. So they really are very much a wild creature, quite different than a dairy cow, for example, or a chicken, or a horse. And an illustration of just how wild honeybees are is they can move right out of a beekeeper's hive when a colony swarms, and the bees will move out into a hollow in a tree, and they'll be perfectly fine living their lives on their own out in that tree, as we shall see. And conversely, I bet there's a lot of beekeepers here that have caught swarms and moved a wild colony of bees in the form of a swarm into one of their hives. So there's a lot of going back and forth. The fact that bees can do that means that they are still quite wild. Now, these bees have been with us. They were not originally in North America. Honeybees, Apis mellifera, is an introduced species. It was brought in starting 400 years ago. I think the first one was 1640 to the Virginia colony, but also into Massachusetts Bay Colony and New Haven and into Delaware. And this map shows a record of the expansion of the bees from these 4 introduction sites as it spread across the eastern deciduous forests. Now, you might be wondering, well, how do you know those dates? How did somebody figure out those dates? There is a gentleman at the University of Cincinnati, Gene Kritsky, who went back through countless diaries and letters of people settling the eastern U.S. and found when they said, well, we found bees in the woods here as they moved into an area. So we actually have inadvertently good records of the expansion of the bee. And it took the bee only certainly less than 200 years, only even less than 150 years to get from the east coast, the Atlantic seaboard, over to the Mississippi River. So these bees swarmed out of the beekeepers' hives and just kept going. And for hundreds of years since then, the wild colonies of honeybees figured very prominently in the lives of people. What we see here on the left is what was done for hundreds of years before the invention of Langstraw's hive. A lot of people got their honey by going out, finding what's called a bee tree, a tree that was hollow where the bees were living in it, and then chopping open the tree or cutting down the tree and splitting it open and taking out the honeycombs. That's very destructive. But I grew up with an old timer. She didn't have much money, and when she wanted a sweetener, she would do this. She'd go out with an axe, a pail, and a ladle, and she was a bee hunter, as I'll explain shortly. She would find the bee tree. She would do as these gentlemen are doing, not killing the tree or killing the colony, but just opening it up, chopping it open. And then with her ladle, she'd scoop out some honeycombs and put it in her pail and bring that home. So the honeybee has long been important to us. And here's just an image from a bee hunter in Vermont. This is where you capture some bees off of flowers, introduce them to a little bit of square of comb, which you filled with sugar syrup, the bees, you've introduced them to a great food source, and then they'll recruit other bees back to where they're going, and you can trail them to their home. We'll talk more about that. What I want to share now is that, these honeybees have been with us here in North America for some 400 years, nearly 400 years. And we now think about honeybees as living in beekeepers' hives. But the theme of this talk is that there are still bees, lots of bees living out in the wild, honeybees living in the wild, and they're very important. And they're actually very important to beekeeping for reasons that we'll see shortly. But the most obvious one is that these bees perform lots of pollination. And here's a little bee. This is a photograph from not too long ago, a bee going into one of our first plants to come out in the spring. But they're important in another way, because they can teach us, and this is probably even more relevant now, or just as relevant, they can teach us a lot about how we can promote honeybee health. Now you might think, well, how does that work? Well, first of all, by their sheer example, as you'll see shortly, they're living out there. Nobody's caring for them. They've got the mites, they've got the viruses, but they're surviving. And we'll be looking at how they do that. And the mite that I refer to is we'll look at it again. We've seen this once already. I show it here, again, to emphasize that it's a pretty small mite, but it's really pretty large compared to the bee. So that's the problem. And the reason that mite is so problematic is that it sucks body fluids out of the bee. It feeds on some of the fluids inside a bee. And in feeding on the bee, either the adult or on the pupil, the developing bee, the pupil stage, they inject viruses into the bees. And it's that spread of the virus, it's called the deformed wing virus, which is really the killer. But the deformed wing virus has been with us forever, but this bee, and I don't have time, with the mite, I don't have time to explain it, but by promoting the spread of the mite, they've actually selected for virulent strains of the virus. And about 40% of managed colonies are dying each year because of the viruses spread by this mite. And here's what that mite does, the virus does to bees. These are mites feeding on a pupa, a mature larva, I should say, of a bee, and that's the adult on the right there that comes out from a cell that's been heavily infested with the mites and the viruses. And you can see it has a number of effects on the bees. The deformity of the wings, hence the name deformed wing virus. The legs are paralyzed, and the abdomen, those of you that are beekeepers can see that abdomen in that bee is kind of stumpy. It doesn't look right. So these bees, they die. They would be a worker, but they will not do any work. And the usual response has been to treat colonies with miticides that will kill the mites. That works at first, but like all insecticides, pesticides, the objects being treated or being killed develop resistance. And most of our miticides, the mites have evolved resistance to them through selection for resistant strains of the mites. Another side effect of that is the honey can be contaminated with the miticides, and the bees themselves can be affected with these miticide pesticides. Both are arthropods, and so bees and mites have a lot of similar biochemistry. So the miticides are not and they're not a long-term response by any means. So plan B is to look to nature, to see what's going on with the wild colonies, which are not, of course, getting treated with the miticides, but are persisting, as we shall see. So what we're going to do next in this talk is we're going to answer these questions. What's happening with the bees in the wild? They're not receiving miticide or pesticide treatments, so have they been wiped out? Another question, if they've not been wiped out, then how come? And if they've evolved resistance to the mites through natural selection, how has that worked? Have they in fact done that? Well, I guess I revealed the answer here, yes. And do they have not only just good genes, but a healthy lifestyle, a good way of life? And the answer there is also yes. And so it's those things, those good genes and good lifestyle, that's what these wild colonies can show us and inspire us to adjust our beekeeping, to do the same thing with our managed bees as we see with the colonies in the wild, or at least as close as possible. Now let's review the timeline. I don't know the timeline of Varroa mite in Oregon, so I have to give it to you for New York State, where I could watch it come in and see what it's done. It entered the United States. This is pretty well documented. First came in Florida in the mid-1980s, early 1980s actually. Reached my hometown of Ithaca, New York, sometime in the early 1990s. All my colonies, and at the time I was keeping about 30 colonies of bees, all of them were laden with Varroa mites in the summer of 1994. They came in cryptically, just a few, but by 94 it was obvious that the colonies had them. And that winter, the following winter, 17 out of 19 colonies died in that 94-95 winter. So no surprise, I began treating my colonies for mites with the miticides in the summer of 1995. And I kept doing that for many years. But in 2002, something very important happened. Sort of a light bulb went off in me. I thought, what about the colonies in the wild? Have they been wiped out? I thought they probably had been, because I had seen so much mortality with the colonies in my apiaries. So I presumed that they were all gone. But I was in a unique situation to actually answer that question with an interesting angle, because in 1978, I had measured the abundance of wild colonies in a research forest that Cornell University owns. It's called the Arnott Forest. This is a landscape view looking down from a satellite at Ithaca, New York, central New York. The land north of Ithaca, where I live and Cornell is excellent farmland. The glaciers smoothed it out. It's excellent soil, limestone soil. But south of Ithaca, the glacier didn't get there. It's hilly, very rugged, very acidic soils. It's not good farmland, and what it grows very well is eastern hardwood trees. And so they're large forest areas. And they're not beekeepers down here. It's not a great place to build big honey crops. And you get a glimpse of what this eastern deciduous forest looks like in October over here on the right. And so Cornell has this Arnott Forest. In 1978, I went to that forest and I looked and I mapped out over 50% of the forest, the locations of wild colonies of bees living in trees in that forest. And this is the map. I hope it's not too small for you I hope it's large enough for you to be able to see it. So this is the outline of the forest. Each of these little black tree things is where I located a colony of bees living wild in a tree. And here's an example of a bee tree over here. This is a hemlock tree, and bees were living up in that hemlock tree, going in and out of this little opening there. So I could find 10 of those searching over half of the forest. And I can't be sure that I found all of the bee trees in that half of the forest. So these numbers I'm giving you of number of bee trees or wild colonies are, if anything, conservative. If you're curious about how you actually find this, it's like finding a needle in a haystack, a bee tree in a forest. You use these old bee hunting techniques of where you go out to a little square of comb, using a little device called a bee box. You capture bees on flowers. You introduce the bees to this comb that's filled with sugar water. The bees find that a marvelous food source. The bees that you've introduced to it, they go home, they perform waggle dances, they bring other bees from their bee tree home to your comb. And in time, if the conditions are right, the weather's good, there's not a nectar flow on, you'll get a crowd of bees on your comb, and you'll see there'll be enough traffic that you can actually see the direction the bees are going home. And then you work your way by moving your little feeding station down the line, 100 yards at a time, step by step, you can work your way back and to the very tree, to the specific knothole. And it's great fun, and you might wonder how long it takes. Sometimes the minimum has been like 35 minutes for me, and the maximum was 3 years to find a bee tree. If you want to know how it could be that widely ranged, I wrote a little book called Following the Wild Bees, where I explain how this all works, and the biology behind it. But anyhow, so in 1978, I had this baseline data on the abundance of wild colonies in a forest before Varroa. So in 2002, I said, okay, I'm going to go back to that same forest. I'm going to go to the same forest, the same season, using the same methods, and I'm going to see if there are still any colonies there. And I expected I would not find them because we had experienced such heavy mortality of the Varroa mites from the deformed wing virus over the previous 10 years. So, 2002, same place, same time, same methods, I found 8 colonies in the Arnott Forest. That surprised me, very much surprised me. But, being sort of a critical scientist, you have to say, well, how come? Maybe these colonies are so remote, they don't have Varroa. As I mentioned, there are beekeepers out here, so I thought, well, maybe Varroa just hasn't gotten out to the middle of these wooded areas. So the way to address that question, I would put up in trees what I call bait hives. These are just old Langstroth 10-frame hives. At the time, that's what I was using. I make the entrance a little smaller. I put them off the ground, mostly to make them hidden from black bears. You can't leave a hive on the ground in the forest. And put those up in May 2003, and 3 were occupied in July, and then I could start measuring the mite levels in those colonies. You'd put a sticky board at the bottom of the hive, and it traps mites that fall down. And what I found was that, I'm not showing you the data for all 3 colonies because I couldn't get to fit on the slide. All 3 colonies were dropping mites. And so all of these colonies had gotten the mites. And now I understand why, even though the colonies were, and it's because of those mites are very good at getting dropped off in a flower and then being picked up by a forager. They don't have to move directly from one hive to the next. They can be picked up by foragers, or they can be spread if a colony dies out with the mites, from the mites, it gets robbed out, the mites glom right onto the robbers and get carried home to the robbing colony's nest. So we now understand why it is that probably every colony in the wild has Varroa mites too, and yet the colonies are still surviving. And because I went in 2011 and I did it again. I measured it, I went back to the forest, and again, I found about the same number of colonies, 9 colonies, searching over 50% of the forest. So these wild colonies in the Arnott Forest are surviving with the mites and the viruses that they carry. So the question is, how are they managing to survive? Good genes? Question mark. Good lifestyle? Question mark. Both? Question mark. Usually at this point I want to check in with folks to see if there's questions about this evidence that the wild colonies have the mites, but they're living. But I can't see people well enough to see if anybody's got their hand raised. But I'll answer one question up front where I can see the arm. Yes?
Well, what is a bad lifestyle for a bee?
The question is, what's a bad lifestyle for a bee? A bad lifestyle for a bee is, as we'll see, is living in a very large colony so that you're a super duper host for the mites. A bad lifestyle is to be treated with miticides by a beekeeper, because that means you'll never get resistance, you'll never evolve resistance for the mites. Another problem of bad lifestyle for honeybees is if you're living in a hive that a beekeeper is preventing swarming. Because swarming, turns out we've learned, creates a break in the brood cycle. There's a period in the colony when there's very little brood, and that shuts off the reproduction of the mites. Another bad lifestyle is to live in a really large nest, because that, again, means you don't swarm, and again, you have a huge amount of brood for the mites to reproduce in. Thank you very much. That was a good warm-up to the rest of the talk. Let's look at the good genes side of things. Do these bees in the wild, do they have some genetic advantages, some genetic abilities to cope with the Varroa? Yes, they do. We know from a genetic analysis, which I don't have time to go through in detail, but I'll share with you the highlights of it tonight. We know that in the 1990s, which is when Varroa reached Tompkins County, New York State, this population of colonies living out in the wild went through a nosedive. They went down to probably about 10% because almost all the colonies were wiped out by Varroa. But there was that approximately 10% of survivors. And those are the bees that have repopulated the forests and show us that bees in the wild can cope all on their own with the mites. What is the evidence for this population crash? The evidence, and this may be hard for you to see, this is a diagram of the family relationships of colonies of bees. This is an inference from looking at what's called the mitochondrial DNA of worker bees. And the mitochondrial DNA is only inherited from mother to daughter. So if you take a worker out of a colony and you look at the mitochondria, you're getting a readout of what the mother queen's mitochondrial DNA tells us. And what we found back in 1977, I'll summarize it because it's too small. Back in 1977, of the colonies I looked at in the wild, I found that there was, in this region of Tompkins County, there were 23 queen bee lineages. There was a lot of genetic diversity. But when I went back and collected again in 2011, did the same analysis, or actually collected, it's complicated, but I had the bees stored from 1977, so I went out until 2011, collected another set. We did the same genetic analysis on them. In 2011, there were only 3 queen lineages in the whole population. And that is prima facie evidence that something knocked down that population, pushed it through a genetic bottleneck. So there was massive colony mortality. And evidently this very powerful selection has produced a survivor stock. And it's not just a good story. People did an experiment in Sweden where they took 150 new colonies of honeybees out to the island of Gotland and didn't treat them, just let them go. And they saw the same thing. In about 3 years, that 150 colonies had died down to about 8. And then, little by little, they left the hives there, let the colonies swarm. Little by little, that population has built itself back up. You might want to wonder, well, what about the other genes, the nuclear genes? That's a whole other story. And there's like over 600 loci that have undergone heavy selection in these bees. We don't know exactly what those genes are coding for, except that we know that half of them are genes involved in development. So they're shaping the bee during development in ways that are evidently benefiting them. Okay, that's the genetic side, but what if these genes change? What about their physiology and behavior? Well, one of the things, and I can only highlight some of the striking things that are better in these wild colonies with respect to Varroa control, is that they're really good at killing the adult mites, biting, uncapping the mite-infested brood cells, and biting the legs off of the adult mites. Now, I know a lot of you are beekeepers. If you put down a sticky board in the bottom of your hive and so you could collect the mites, you can do this. It's very easy to do. And then if you painstakingly pluck off those Varroa mites, just put cooking oil on a piece of cardboard or something, and you look at how many of them have missing legs on them, it'll probably find, if you're buying your queens from California or Hawaii or Florida, it's probably going to be about 5 to 10% of the mites have missing legs. But if you get them from this Arnott forest or Gotland or one of the other places around the world where natural selection has acted, you're going to find about 50% of the mites are missing legs and are thus die. Mites die after the legs have been removed, or a leg has been removed. But there's another trait that kills the adults. But what about disrupting the Varroa's reproduction in the brood cells? Well, these wild bees are very good at that too. And those of you that are beekeepers probably know about this. It's called, it's assayed using an assay called the freeze-killed brood assay. This is where you take a frame that's got sealed brood, capped brood. You put a ring on that, about 4-inch diameter ring. You fill it with some liquid nitrogen. It freezes the brood. It kills the brood. And then you put after the liquid nitrogen has evaporated, you put the brood frame back in a hive, and you come back 48 hours later, and you see how many of those cells that had dead brood in it have had the brood removed. And if the colony is very good at this freeze-killed brood assay, and thus is very good at removing dead and dying brood, you'll get the image shown on the left here. Almost all the cells are open. And that's what you get if you look at a frame from a colony that has a queen that's from the Minnesota hygienic line, or some of the breeders now are incorporating this into their selection. It's also what you find with the Arnott forest. Virtually 100% of the cells are cleaned out. But if you get unselected bees, i.e. ones from most queen breeders, like in Minnesota or Australia or New York, it'll be down around 30 to 40%. So that's a big difference. And that with so many cells not being opened up when the brood is dead or dying inside there, that means the mites can reproduce. So are the wild colonies surviving thanks in part to good genes? Yes. Another aspect, which I'll just mention it in passing, is that the development time of the wild bees is shortened a little bit. And that means makes that a little bit harder for the Varroa, too. They don't have as much time to develop. But what about lifestyle? The answer here is yes. These wild colonies also enjoy at least 20 lifestyle advantages. Now I feel a little bit like one of those late night TV show people. Alas, I do have a book on this subject. But anyhow, we won't go there. What are some of these lifestyle advantages that these wild colonies benefit from? Well, what are some of the differences? The wild colonies, this came up with the gentleman's question earlier on, so this is kind of like a review. The wild colonies are widely spaced. They're on average about a half a mile apart instead of a half a foot apart. And this means the spread of the pathogens and parasites is reduced, particularly Varroa. And we see this particularly, we're learning now that, I mentioned this once already, but I'll emphasize it, I'll repeat it to emphasize it. One of the most powerful ways these mites are spread around is when a colony collapses, it loses its defenders, and bees from nearby go in and rob it out. The colonies that do the robbing, their mite counts shoot up very quickly. So being spread out in the wild, they don't get robbed out as quickly or as frequently when there's a nearby colony that has succumbed to the mites. Another difference in lifestyle is that these wild colonies occupy nest cavities that are only about the size of one level of a 10-frame Langstroth hive. That's about 40 liters. And what is that? And so here we've got a slide that shows just a distribution of the nest cavity size in bee tree nests. And I've measured it here in liters, but more usefully for beekeepers in units of number of deep hive bodies. 1, 2, 3, for example. So bees in the wild live in a hive that's about like a little bit smaller, a little bit bigger than just one deep Langstroth 10-frame hive body. They're not living in big skyscrapers like we see there on the right. And what does that mean? It means that the colonies in these small nest cavities, they have a lot going for them with respect to the mites. They swarm frequently, virtually almost at 90%. 0.9 probability of swarming each summer. It also means, and that means that there's a break in the brood rearing. It also means that because the nest is small, they start to plug up the brood nest, they start to plug up the cells in the nest with honey in my area in late August. So you get into September, there's no brood for the Varroa to reproduce in. So that also inhibits the Varroa reproduction, living in these smaller colonies. Fewer cells of brood, more breaks in brood rearing, and termination of brood rearing early. A third lifestyle thing that's going on in the forest that's different in the apiary is the wild colonies coat the walls of their nests with propolis. Here's a bee carrying home a load of propolis. And here's what they do. This is the floor of a wild colony nest. It looks like linoleum has been laid down in the floor there, but it's the same thing you would see on the walls. And the bees are stimulated to do this because when you've got a decayed tree cavity, they will remove the soft, decayed wood, but they're still left with a surface which is very porous. And that's like the ideal substrate for bacteria and molds to grow on. So the way they cope with that is they plaster it over with these antibiotic tree resins. And there's some recent work that's been done that shows that not only does that prevent bacterial growth and mold in the walls of the nest and the floor of the nest, but it also shows up in effects on the bees' immunity genes. People have looked at this and they find that if bees live in a hive where the walls are coated with propolis like they are in a bee tree, the bee's immunity genes are not working. They're not activated to the same level. And that means the bee is investing less, and this is particularly true for the nurse bees. That means those bees are not using up resources, just fending off infections. And so they've got more resources to produce food for the brood and do all the brood care. Better nutritional state of the nurse bees, thanks to the propolis. And just one more thing I'll mention that's a lifestyle advantage of the bees in the forest is they occupy very well insulated nest cavities. Managed colonies do not occupy well insulated nest cavities. This is something we've we have forgotten about and overlooked in our beekeeping. I didn't realize this, but if you go back to I did realize this, but I brought it back to my memory. When I started beekeeping in the 60s, the old timers, they would really pack their bees for the winter. In fact, they put them in packing cases, big cases, and then they put insulation, straw and sawdust, and closed the whole thing up. Those bees were very well insulated to get through the winters. Now, we beekeepers just put them out in the hives, and we might wrap them with tar paper. But a lot of our hive design now and our whole management was with thin-walled hives is based on migratory beekeeping. It's pretty easy to move the hives we have around. But in the wild, they don't move around at all. They're in these big, thick tree cavities. And to look at the effects of having a very thick tree cavity, thick walls of a tree cavity, I made a tree cavity of a very specific size with an electric chainsaw. And it was 40 liters, and then I put a door back inside, and then, so it's here, it's sealed up. I could put temperature monitors inside, could record that, and then for reference, I built another hive, identical in dimensions, and entrance opening size, and even the color was as matched as I could make it. And what I found was that there's a world of difference between the temperature climate, the microclimate inside a cavity like this and inside this box, like it's a beehive. This blue line shows the temperature records over a string of days in April inside the tree cavity. It's very stable. And these jumpy ones are either the yellow is the upper temperature reading in the box, and the black is the ambient. So the temperatures inside the box are almost jumping around almost as much as outside the box. There's just not much in those walls. So that's a big difference. We don't know fully what the implications of that are, but I suspect it means that the bees are having a much easier time in a well-insulated home. We used to think that the problem with insulation was that putting lots of insulation would be that the bees would get lots of moisture. But what we're finding is that when the bees live in a very well-insulated hive, there still is condensation in the hive, but because the nest is well-insulated, there's a warm pocket in the top of that cavity. It's so warm up there that the surfaces the temperature is not below the dew point. There's no condensation up in the upper part. The condensation is below the bees. And we've also learned that in the winter, at least where we are, where they can't go out very often, they use that condensation as a water supply in the winter. They get very, very thirsty without that, if they don't have that water in the hive. So we're learning a lot of curious things that are just looking at how they work in the wild. We're learning, it gives us a very different picture of life in a bee colony. So I don't want to take up too much time, so I'm going to end here, but I'll stress these take-home messages. First, the honeybee Apis mellifera remains a wild animal. It lives just fine on itself out in the wild. And we see that. It moves easily from beekeepers' hives out to the wild and then back into the hives. The populations of wild colonies are good sources of survivor stock. I don't know, I'm limited really to the eastern deciduous forest area, but I know a lot of beekeepers in Maine, New Hampshire, Vermont, New York, Pennsylvania. That's how they get their bees. And in Missouri, Leo Sharashkin, that's how they get their bees, and they do well without treatment. But you have to keep them in little hives. They have to let them swarm, but they have a lot of good traits going for them. And then the third point is that and this is the fundamental one, how the bees live in the wild can guide us to improve our methods of beekeeping. This isn't going to work for a big commercial beekeeper, but for a hobby beekeeper, you can implement a lot of these insights that we're learning from the wild bees. Improve our methods of beekeeping so they harmonize better with the bee's natural history. Better insulation, smaller hives, more widely spaced hives, rougher inner walls, allowing swarming. Again, this is not a recipe for a commercial beekeeper, but it is a recipe for the small-scale beekeeper who maybe wants to relate to the bees less like somebody that is keeping chickens to make lots of eggs, and instead is a bird watcher that's just putting up birdhouses and enjoys having the birds around their house. That's what this kind of beekeeping that I'm referring to is Darwinian beekeeping. That's what it's most relevant for. But it's another way to do it, and I hope with this talk you can see that why I think it is valuable to look to these wild colonies. And I think I would summarize the main message of this talk by saying that as beekeepers, we've never really known what we were doing with the bees until we understand what we've been undoing to the bees through our beekeeping techniques. So I thank you for your interest.
This is now your time to ask questions. I will run around with the mic. We will try to hit as many people as we can. I'm coming to the red jacket to begin.
You have good night vision, Amber.
How are the mites evolving?
Yeah. There's evidence that mites are evolving to be ever more virulent. But, I don't think I'll ever evolve resistance to having their legs bitten off. or having the brood cell opened up and the infested pupa taken outside. So I think the bees will stay ahead of the mites on this. Time will tell. Well, time has told us the colonies are living in the wild. The bees are getting the upper hand on the mites. And I want to stress that. It's not just the Arnott Forest. It's not just Gotland Island. In France, there's a beekeeper who can't find mites on his bees. He hasn't treated them. We're finding it in Pennsylvania. We found it, where was another? A neighbor of mine looked in another forest nearby, and he's been capturing his bees out of that forest, and he keeps close track. Anyhow, I should stop talking. A question?
So I've loved the lecture so far, especially because of the emphasis on Varroa destructor. This is a fairly new idea to me, yes. Although when I read about colony collapse disorder, the things that I've been reading about still widely promulgated are pesticides like neonicotinoids is one group. And then I've often wondered about other pesticides as well. There's one called Termidor, which is used to fight termites, which is also very destructive to bees. And I wonder what your view is on what role pesticides might play in collapsing colonies as well.
Yeah, thank you for asking that question, because pesticides really can be a big problem separate from Varroa in many locations. Where if you're in an area where there's where your bees are going into crops that are sprayed with pesticides, then yeah, that's another challenge. That one is, in some ways that's a little maybe easier because we control what those pesticides are and how they're used. So and there are ways for, I know at least where I grow apple orchard, people to do their spraying in ways that minimize the damage to the bees, particularly doing it even at night or very early in the morning.
All right, we're in the same aisle over here.
Hi, I was curious about the impact of honeybees on native bee populations. Mm-hmm. Is this something you're concerned about?
Yeah, I do have concerns about the impact of honeybees on native bee populations. And my concern mainly is not in areas where people have had the honeybee in the area for 400 years, but there are places out west here in the Rocky Mountains where there are high desert areas where there have not been honeybees, but some of the large commercial beekeepers pulling their bees out of the almond orchards are looking for good forage locations up in the mountains. And when they come in with a tractor trailer load of honeybee colonies, those honeybees can really soak up the resources in those valleys, and that really could have an impact. So I do have concern about that. And usually that's U.S. forest land, and they are developing a policy to protect that.
All right, I know we could ask questions all night, but let's give it a round to Tom Seeley.
Thank you.
Thanks for listening. If you've liked what you've heard, I'd like to ask a favor. We're still trying to get the word out about the podcast, and it would be super helpful if you'd follow us on social media and tell your friends. We're on Facebook and Twitter at SciBar Podcast. That's SCIBAR Podcast. If you're in the area and would like to come to an event, our list of upcoming topics is at our website at scienceontaporwa.org. And that last part stands for Oregon and Washington. You can also find out more about the events on Facebook, Twitter, and Instagram at scienceontaporwa. As always, I'd like to say a big thank you to my volunteers who I affectionately call the Minions. They are Scott Fry, Chris Gowan, Sam Lauk, Rita Nygren, Steve Perry, and Chris Trone, as well as a lot of other people who've helped throughout the years. These events and these podcasts would not happen without them. I'd also like to say a special thanks to Amber Peoples for hosting this event and for running things for the past few months, and to Graham Tully for audio production. Also, a big thank you to Jonathan Coulton for letting us use his song Mandelbrot Set as our intro and outro music.
Badass fucking fractal, and you're just in time to save the